Thank you. Hello, everybody. My name is Andrew Tutt at the Institute of Cancer Research in King's College in London. This is a great honour for me to be invited to give a plenary lecture at GBCC, and I have not done this before, so it's a very, very great honour for me. Thank you so much for this opportunity, and I hope that I can provide some really helpful and interesting information for you. Yes, indeed. I was privileged to be invited to Korea to speak at the KSMO meeting a few years ago before the pandemic. And I was aware of the conference then and many of the organisers, and sadly have not been able to participate until now. But I'm very well aware of it and very well aware of the contribution of Korean colleagues to breast cancer research, some of which I'm going to talk about that contribution in my lecture. Of course, thank you. So I am going to be speaking about the role of some systemic therapy developments in early breast cancer in patients who inherit germline mutations in the genes BRCA1 and BRCA2. We know so much more now about the biology of these genes, and myself and others working with international collaborators, many in Korea, have been trying to develop targeted approaches to therapy for women who develop breast cancer in the context of having these faults in the BRCA1, BRCA2 and some other genes. And so I'm going to speak about the history of the development of those approaches and recent trial results in early breast cancer that are looking at PARP inhibitors and to some extent the targeted use of chemotherapy platinum drugs in this population of patients and update information on overall survival results from a key trial in which Korean collaborators have been really engaged, the Olympia trial. I will also go on and explain a little bit more around other trials in the field in early breast cancer and where new work is going to try and go beyond what we currently know from trials like Olympia and try and explore areas that we don't yet have the answers to. Try to integrate the information from the trial with other emerging standards of care in breast cancer that is relevant to those who have BRCA mutations, but are not specifically targeting those mutations. So new therapies relevant in ER-positive and triple-negative breast cancer that remain relevant in those with germline mutations. And then I'll speak a little bit more about how we might target and even possibly prevent the development of resistance to some of these BRCA-directed therapies like PARP inhibitors in the final part of the talk. Well, thank you. It's very flattering to be asked to advise sort of more junior colleagues from a career perspective. I'm not sure I've got it all right. I've certainly enjoyed my career to date. I think, and my advice would be really take the opportunity to link the amazing amount of non-medical science in cancer research to the needs of your patients. I think care to try to understand where their problems in their care of cancer could be addressed not just by clinical trials, but by explaining those problems to cancer researchers in the labs. And then you can work together to try and solve those problems in the clinic, design trials together with lab scientists and develop new treatment approaches. And that keeps your career really exciting and fresh and trying to take the field forwards. And I think cancer and breast cancer specifically is a really good opportunity to understand those areas and have a really rewarding career. I would try in general to say to people, find people that you really enjoy working with and trust working with, and then keep working with them. Find new problems to address with that team of people, because it will be rewarding, it will be productive, and you don't always need to find new people to work with if you've developed an effective team. I guess that would be another key piece of advice. And really value translating the problems of the clinic into the research world and translating back from the laboratory, if you get the chance to spend some time in the lab, translate between the two, because that's where you will make the biggest changes to the treatment of patients, and it's a really rewarding thing to try to be a translator. Well, one thing I'd like to suggest that we might try to remove a word or remove a phrase by the research we do. So I would like us in 5 years' time not to be talking about triple negative breast cancer. I would like us to have taken that group of diseases that are negative for a number of receptors and turned them into some positives. So I think we've done that with BRCA mutation positive triple negative breast cancer. We have a treatment approach now for that group. I would like for us to get rid of the term triple negative breast cancer because we've replaced it with positive biomarkers we can work with. I think the other key words I think are going to be around biological therapies in breast cancer. I think there are some very smart new ways of using antibodies, antibody drug conjugates we're hearing more and more about, specifically engineered antibodies to engage the immune system, and cellular therapies, CAR T-cell approaches or combinations of vaccines with checkpoint inhibitors. So I think cell therapies and biological therapies are going to be key words. Digital pathology, the use of spatial analysis to look at how tumours are a complex mixture of cancer cells and the microenvironment, and using digital pathology and spatial analysis to work out prognosis better and where new therapies will have most effect is going to be a key word going forward. And I think plasma biopsy, I guess, is another phrase. We're learning so much more now from the study of plasma, a lot from DNA, but I believe we will find other things that we can understand from the patient's plasma during their treatment that will inform how best to treat them. It's thank you, and it's thank you generally for the invitation to be at the meeting and to speak at the meeting, but it's also a specific thank you to the many who have been fantastic collaborators in the Olympia trial in Korea, which has been a country that has made a huge contribution to this important international collaborative trial in a relatively small subgroup of breast cancer patients with genetic disease, and which needed real effort across the globe. And Korea made a huge effort. And I want to say thank you to all of those patients and their doctors and the lab teams involved. So thank you very much.